Hello everyone, thank you very much for joining the webcast today. I am Mayumi Fujita, Senior Researcher of National Institute of Radiological Sciences and National Institute for Quantum and Radiological Sciences and Technology in Japan. So today I'd like to talk about the distinct metabolic profiling of invading PANC-1 pancreatic cancer cells. So here is the outline for my talk today. There are 4 parts, and the first part is the background. And I'm going to explain about the cancer cell invasion and transwall invasion assay. In the second part, I'd like to show you the IncuCyte Live-Cell imaging for identification of invading cancer cells. And in the third part, I'm going to show you the metabolome analysis of PANC-1 invaded cells. And the last part is the conclusion. So here is the background. During the metastatic stage, cancer cells invade through the tumor tissue and move into the surrounding stroma, then intravasate into blood vessels or lymphatics. But as you can see in the diagram, not every cell invades and metastasizes. And only those cells which have acquired an invasive phenotype can get into the surrounding stroma and metastasize. First, understanding the characteristics of such a distinct tumor cell population, especially exhibiting the invasive phenotype as compared with the non-invasive cell population, is really fundamental for developing the novel strategies to counter metastasis. So then how these cancer cells invade and move through the surrounding stroma? The stromal environment of cancer in vivo actually predominantly consists of networks of type 1 collagen, like shown in this diagram. So from this diagram, we can imagine that the invading cancer cells should have the ability to move through such a complicated extracellular matrix like collagen networks. And the recent studies have shown that invading cancer cells actually use a variety of ways to move through ECM networks, either by breaking the networks with proteases, moving through the ECM by forming the leading edge at the front and the lagging edge at the back of the cell body, or by migrating through the ECM as rounded bodies, forming a bleb-like protrusions without breaking the ECM. So again, cancer cells exhibiting the invasive phenotype should have the special ability to move through ECM. And in order to study the characteristics of the distinct tumor cell population that's specially exhibiting the invasive phenotype, we first have to determine and figure out which cell lines have higher invasive potential. And this diagram shows the commonly used experimental setups for determining the tumor cell invasiveness in vitro, which is called the transwall invasion assay. And this is a really popular assay model to measure the invasiveness of cancer cells. Okay, there are 2 media in the chambers. Usually serum-free medium is placed in the upper wall of the transwall, and the serum-containing medium is placed in the bottom wall. And then they are separated by porous membrane on which a thin layer of ECM is coated. And as you can see in the diagram, only the cells that have the invasive phenotype can move through ECM and reach the underneath of the transwall membrane. So by staining those cells reached underneath of the transwall membrane, we can count how many cells have actually invaded through the ECM from the total number of cells placed on the upper wall of the transwall membrane. So that, for example, if we use several cancer cell lines and then performed this invasion assay for each cell lines independently under the same assay condition, we can figure out which cell line is the most invasive and which cell line is the least invasive. Then we can choose the highest invasive cell line and the lowest invasive cell line, and then use those cell lines to compare and examine the factor affecting such different invasive ability, like examining the difference in protease expressions or difference in cytoskeletal regulation and so on. And in our experiment using 4 human pancreatic cancer cell lines, as you can see in this figure, MIR-PACA2 was the most invasive cell line, and then SBC1 was the least invasive cell line among these 4 cell lines. So that we can just simply choose MIR-PACA2 and SBC1 cell line to compare the differences. However, interestingly, when we count the percent of invaded cells in this assay, we found that only a few percent of cells invaded through ECM. Like in the case of MIR-PACA2, only about 3% of cells in the whole cultured MIR-PACA2 were able to invade through ECM. And in the case of PANC-1, only about 1% of cells in the whole cultured PANC-1 were able to invade through ECM within 24 hours. Furthermore, if we extended the time of invasion assay for MIR-PACA2 or PANC-1 to be 48 or 72 hours instead of the 24 hours, surprisingly, the percent of invaded cells were still almost similar as the one counted at 24 hours. So this is suggested that they might have the distinct cells, especially exhibiting an invasive phenotype within the whole cultured MIR-PACA2 or PANC-1. And actually, it is a really interesting point because MIR-PACA2 or PANC-1 is just the identical cell line, which means that every cells in whole cultured MIR-PACA2 or every cells in whole cultured PANC-1 should have the same genetic background. But actually, the data showed that only a few percent of cells within the whole cultured MIR-PACA2 or PANC-1 were able to invade. And if there are such a distinct population exhibiting an invasive phenotype within the whole cultured cells of the same cell line, it would be interesting to collect these distinct invasive population and study the characteristics conferring the invasive phenotype with comparing to the whole cultured cells, which have the same genetic background. Like metabolites consumed in different cell lines, for example, are thought to be affected by their different genetic backgrounds, so that in order to determine the unique metabolic profiling of invasive cells, collecting the distinct cell population exhibiting the invasive phenotype from the cells of the same cell line would be intriguing. But is it really true that there are distinct cells, especially exhibiting an invasive phenotype within whole cultured MIR-PACA2 or PANC-1? To identify this, we used the IncuCyte Live-Cell Analysis System and capture the movement of the distinct invading population in real time from within the incubator. This would minimize the disturbance to the cells and allowed us to collect the kinetic information. So we made a spheroid model using MIR-PACA2 or PANC-1 cell lines and embedded in the matrigel. And the cells from the spheroid that embedded into the matrigel were visualized with the IncuCyte Live-Cell Analysis System. So here are the procedure. Cells were seated into the 96-well ultra-low attachment plate with 100 microliter media, and spheroid formation was preceded by incubating the plate in the CO2 incubator for 3 days. Then half of the media was carefully removed, and 50 microliter of matrigel with concentration of 6 mg per ml protein was poured into each well, so that the final concentration of matrigel was 3 mg per ml. And then this was solidified for 2 hours in a CO2 incubator. After that, we carefully set the plate into the IncuCyte zoom and captured the movies of the real-time imaging of the spheroid embedded in the matrigel. So this is the movie for PANC-2. This is really a beautiful movie. So you can see that actually only the distinct population within the spheroid can invade into the matrigel. And also we can see that precise information of the way of PANC-2 invasion, like it forms elongated shape to invade through matrigel. Okay, this second movie is for the PANC-1. Again, you can see that only the distinct population within the spheroid can invade into the matrigel. And also, we can see that in contrast to the PANC-2, PANC-1 actually formed the rounded morphology to invade through the matrigel. So from those movies, it is clarified that there are distinct cells that specially exhibiting an invasive phenotype within the PANC-2 or PANC-1 cell line, so that it would be intriguing to collect such an invasive cell population and study the characteristics conflicting this phenotype with comparing whole cultured cells. But then, do every cell lines have those distinct invasive cell population? Actually, the answer is no. For example, SF126, which is a human glioma cell line, showed that there are no distinct population of invasive cells, and every cells of the spheroid exhibited high invasiveness, and they all move outward from the spheroid. So in the case of this kind of cell line, it is better not to proceed the comparison study of invasive versus non-invasive cell population within this cell line. And we can say that before proceeding this kind of study, it is better to first perform the IncuCyte Live-Cell imaging to make sure whether there are a distinct invasive cell population within the cell line that you are using, so that you can get the idea whether the cell line that you are using is good for the collecting the distinct cell population or not. Okay, so this is the summary for the second section. So using IncuCyte Live-Cell Imaging System, we could visualize a beautiful movie of the actual invading cells from the spheroid, allowing us to identify the distinct invading subpopulation from the cells forming the spheroid. So in the next study, we then analyzed the unique characteristics conflicting this invasive phenotype with using the PANC-1 cell line. So this is the third part, and in here we performed the metabolome analysis of PANC-1 invaded cells. Invasive cells must alternate their ECM adhesion state and display directional polarity and modifying their shapes. So in contrast to the non-invasive cells, it is hypothesized that the invasive cells need additional energy sources and upregulation to move, such as amino acid, nucleic acid, lipids, or fatty acid. So from that, we hypothesize that the invasive cells may show the unique features in the metabolome. And as I mentioned in the previous slide, the metabolites consumed in the different cell lines are thought to be affected by their different genetic background. But if we can collect the distinct invasive cell population from the cells of the same cell line, it would be intriguing for analyzing the metabolism, especially relevant to the invasive phenotype, because we don't have to care about the difference in the genetic background. So in this study, we used a PANC-1 cell line, and we collected the embedded cells as we identified using the IncuCyte, and compared the metabolomic profile with those of whole cultured PANC-1. The procedure is summarized here. So we first confirmed that there are the distinct invasive population within the whole cultured PANC-1 with using the IncuCyte Live-Cell Imaging System. Then we performed the transport invasion assay with PANC-1 and then collected the invaded cells reached underneath of the transport membrane with the treating cells with actase for 30 minutes on room temperature. The control group was a dish of cultured PANC-1 cells. We used this as a control because non-invasive cells in the top of the transport stayed in a serum-free medium for a long time, so that the medium condition is different from those of the invaded cells. Also, the invaded cells are only about 1% of the PANC-1 cells. So, which means that 99% of the whole cultured cells are non-invasive. So we use whole cultured cells as the control compared to the purified embedded cells. Okay, here are the results of our metabolome analysis of the PANC-1 invasive cells. PCA indicates that the metabolome profile of the embedded cells was very distinct from those of the whole cultured PANC-1, suggested that invasive cells have the unique characteristics in their metabolome. So this is the diagram shows the differences in sugar metabolism for the invasive versus control cells. The glycolytic intermediate of the invaded PANC-1 showed both a reduction and increase in their concentrations as compared with those of control. For example, the level of glucose 6-phosphate was reduced in the invasive cells, whereas the downstream metabolites 3-PC and PEP were both increased. In addition, lactate levels were decreased in the embedded cells, suggesting that lactate fermentation was reduced in the embedded cells. Recent studies have reported metabolic changes during the progression of metastasis. Like Liu et al. Reported in JBC on 2010 that they compared the metabolome of the normal mouse mammary epithelial cell line to those of an isogenic series of cell lines which is exhibiting higher metastatic potential in vivo. And in that report, they found that the glycolytic intermediate, especially 3-PC and PEP, and pyruvate were upregulated in the metastatic cell lines, as we observed in embedded PANC-1 cells in this study. So since invasiveness is one of the more important abilities to metastasizing cells, increasing levels of 3-PC and PEP may be one of the unique metabolic features relevant to the invasive potential. So then what happened to the cells when their 3-PC and PEP were upregulated? The specific accumulation of the 3-PC and PEP was observed when cells redirect glycolytic carbon into the serine and glycine biosynthesis pathway. There is a biomedical evidence that the phosphate group from PEP can be transferred to the histidine residue of PGAM, where phosphorylated PGAM is thought to increase the ratio of 3-PC to 2-PC. And it makes the 3-PC and PEP in particular accumulated. And we have not yet determined the phosphorylation levels of PGAM nor the activity of serine synthesis, but it would be intriguing to further study whether the specific accumulation of 3-PC and PEP observed in the invaded PANC-1 were the result of glucose flux directed to the serine synthesis or not. Okay, this figure shows the TCA cycle. For the TCA cycle, cyclic acid, succinic acid, and malic acid were all reduced in the cells with the invasive phenotype. Thus, those metabolites may have been utilized more than they were produced in invasive cells, or they were simply consumed less in the invasive cells. We also calculated the adenyl energy charges by using the formula indicated by Atkinson et al. So AEC score of most cells are known to range from 0.8 to 0.95. And if AEC score showed higher number than this, it means that the downregulation of energy metabolism. But if AEC becomes lower number than this, then it means that upregulation of energy metabolism. And from our result, AEC score was significantly lower in the invasive cells compared with the whole cultured cells at 0.69 for the invaded cells and 0.82 for the whole cultured cells respectively. So that it is indicated that invasive cells consumed more ATP than the whole cultured PANC-1. And accordingly, this suggests that ATP generating pathways were stimulated in the invaded cells as it was indicated in the Atkins paper. Next, we calculated the indicator of arginine metabolism. Because there were the differences in the amino acid relating to the arginine metabolism, and it is known that arginine is metabolized to stololin by NOS, but arginine also can be metabolized to be ornithine via arginase. So the ratio of stololin to ornithine can be the indicator of the consumption of arginine by NOS versus arginase. And the amount was higher in PANC-1 invaded cells, so that it suggests that invaded cells use NOS more than arginase to metabolize arginine. In addition, the ratio of stololin to arginine, the indicator of NOS activity, is also higher in invasive cells. First, this suggests that the invaded cells are producing more NOS than the WCC. Interestingly, we have already reported the role of NOS and PANC-1 invasion in the recent study, and the treatment of PANC-1 with a NOS inhibitor diminished the invasiveness, whereas addition of NO donor increased their invasiveness. So it suggests that higher activity of NOS required in the invaded cells caused higher arginine conversion to be stololin, and as a result, the unique profile of arginine metabolism was found in the invaded cells. Glutathione is one of the major factors involved in antioxidation. Intracellular glutathione usually exists in its reduced form GSH, and GSH is converted into the oxidized form GSSG by stimulation with oxidative stress. So when cells are exposed to oxidative stress, GSSG are accumulated, and the ratio of GSH to GSSG will decrease. Therefore, the ratio of GSH to GSSG is a useful indicator of the oxidative stress in the cells and has been used as the index of the intracellular oxidative stress. So, in this study, we also used GSH and GSSG concentration from the metabolome analysis and calculated their GSH-GSSG ratio. And actually, we found that the ratio of GSH to GSSG was significantly lower in the invaded cells compared with the whole cultured PANC-1. So it suggests that the invasive cells may have experienced higher oxidative stress than the whole cultured cells. And interestingly, consistent with these findings, Piskonova et al. Also reported in Nature on 2015 that metastatic melanoma cells showed lower values for the ratio of GSH to GSSG. In that study, they used tumor-bearing mice to compare the metabolites of subcutaneous tumors with those of circulating tumor cells or lung metastasized cells, and reported that circulating tumor cells and metastasized cells were experienced higher oxidative stress and showing lower GSH to GSSG ratio than those observed in the subcutaneous tumors. So from that study, they concluded that melanoma cells with especially resistance to oxidative stress can successfully metastasize. So then how about in the invaded PANC-1 cells in our study? The greater production of oxidant result in higher oxidative stress to the cells, which then impair cellular functions. So we next hypothesized that invaded cells may be more resistant to the oxidative stress, as Piskonova found it in circulating tumor cells and metastasized cells. Third, we next compared the sensitivity of invaded or whole cultured PANC-1 to oxidative stress by performing the cell surviving assay with the addition of H2O2. And here is the result. The graph shows the survival fractions of whole cultured PANC-1 or invaded PANC-1 after exposure to H2O2 for 24 hours. The data shows that the surviving fraction of invaded cells for 450 micromolar or 500 micromolar of H2O2 exposure was significantly higher than those of whole cultured cells, suggested that invaded PANC-1 cells were more resistant to oxidative stress than the whole cultured PANC-1. Furthermore, we used BSO, which is an inhibitor of GSH biosynthesis, to see the effect of low GSH on the invasive capacity of the PANC-1 cells. Treatment of PANC-1 cells with 0.3, 1, 2, 3, or 4 micromolar of BSO for 24 hours clearly reduced the intracellular GSH content in a dose-dependent manner. However, it is also toxic to the cells. So as you can see, for 2 micromolar of BSO, the number of live cells were drastically reduced. And when we performed the invasion assay with the BSO, interestingly, although 0.3 micromolar of VSO or 1 micromolar of VSO diminished only 8% or 11% of cell numbers, these compounds inhibited PANC-1 invasion more clearly, like 23% or 46% reduction respectively. So it was indicating that GSH actually has a role in PANC-1 invasion. So this is the summary of this whole study. In this study, we demonstrated that the metabolic profiles of the invaded PANC-1 cells were distinct from those of the whole cultured PANC-1. And in contrast to the whole cultured PANC-1, the invaded PANC-1 cells were characterized as the population with increased consumption of ATP, with assumed activation of ATP-generating pathways, and higher arginine utilization by NOS. And although they had higher oxidative stress, the invaded cells were also more resistant to it, with greater survival upon exposure to H2O2. And reduction of intracellular of GSH by BSO inhibited PANC-1 invasiveness. So this is the conclusion of the presentation. From this study, again, we found that the unique metabolic characteristics for the invasive PANC-1 cell population using the IncuCyte Live-Cell imaging at first. And from that information, we could develop a way to reduce the invasive capacity of the cells by using BSO. So seeing is believing. This IncuCyte Zoom system can give us a lot of important information about the cancer cell invasion, and such methodology is readily available for similar assessment of invasive cell phenotypes. And this is the last slide showing acknowledgments. Great thanks to all colleagues from Advanced Radiation Biology Research Program from National Institute of Radiological Sciences in Japan, and Cancer Stem Cell team from National Institute for Quantum and Radiological Sciences and Technology in Japan for helpful discussions and advice. And great thanks to all colleagues from Dr. David A. Wink Lab from NIH for providing expertise discussions. And great thanks for amazing real-time imaging system IncuCyte Zoom from Zeritorius company. And great thanks for metabolome analysis from Human Metabolome Technologies in Japan. So this is the end of the presentation. Thank you very much for attending this webcast.
Thank you for your presentation, Mayumi. It is now time for the Q&A. To ask Mayumi a question, just type it in where it says type your question here and then press submit. So our first question, Mayumi, and this one asks, can you describe in more detail the method you use to collect the invaded cells from underneath of the transwell membrane?
Okay. Thank you for the question. So first of all, we have to use the large size of transwell insert, which just fits the 6-well plates. And then we coated the transwell with matrigel, as I mentioned in the slide. And after the solidification of the matrigel, we seeded 1 million of PANC-1 cells on the upper wall of the transwell and performed the invasion assay for 24 hours. And then we wiped off the matrigel with a cotton swab so that the non-invaded cells stays on the matrigel, or the stays inside the matrigel were all wiped off. And then we washed the transwell with PBS for 2 times. So for washing, we simply put transwell into the other well of the 6-well plate, which is filled with PBS, and then leave the transwell in PBS for 3 minutes for 2 times. So we can wash the underneath of the transwell with PBS for 2 times. And then after that, we put transwell into the new well of 6-well plate, so which is containing the active, so that we left that transwell for 30 minutes at room temperature inside the active. And then during that time, cells attached underneath of the transwell membrane were detached from the transwell membrane and then suspended in the active solution, so that by collecting the active solution, we can finally collect the invaded cells. And also there are another important thing that, as you know, that invaded cells were only like a few percent within the whole culture cells in the case in PANC-1 cell line, so that we have to use like hundreds of transwell to collect the invasive cells. So that's the really hard work part.
Excellent. Thanks, Mayumi. Our next question is from Fernando, and this one asks, have you seen other signs of higher oxidative stress in INV cells versus the whole cells?
Can you say it again?
Yeah, sure. So Fernando was asking whether you've seen other signs of higher oxidative stress in the INV cells versus the all of the cells.
Okay. As I showed in the slide, I did the oxidative stress experiment for comparing invasive cells and then whole cultured cells, and with using the H2O2, addition of H2O2, and then actually invasive cells shows more resistance to the higher amount of H2O2. So they are actually the resistance to the oxidative stress than the whole culture cells.
Excellent. Thanks, Mayumi. Our next question, and this one is from Seema, and this one asks, how do you know that the INV cells have a similar genetic landscape as the non-INV cells? And have you then performed any next generation sequencing analysis for these 2 populations?
Okay, this is, thank you very much. This is a really good question. Okay, so at the first point, I collected the invasive cells from the identical cell line PANC-1 itself. So they are all PANC-1, the part of the PANC-1 cells. So every cells were from PANC-1, so they have the identical cell line, identical genetic background. So, but I have not actually determined whether they have exactly the same genetic background or not. But I think I also am interested in that invasive cells might have I think that it's intriguing to study the possible involvement of epigenetics in the regulation of invasive cells, and then compare how there are differences in epigenetics between invasive cells and whole culture cells.
Excellent. Thanks, Mayumi. Our next question is from Ahmed, and he asks two questions. So the first one is, how was IncuCyte useful to the 6-well invasion assay? And his next question is, did you try to grow any of these invasive cells and characterize them after a few passages? So is it possible for you to answer either of those two questions?
Okay. I actually don't get the idea of the first questions. Like, I do the invasion assay with the 6-well type of transfer, but it's different from the IncuCyte Live-Cell Imaging. So they are the different experiments. So before using the cell line for the transfer invasion assay, I actually performed the IncuCyte Live-Cell Imaging to make sure that whether they have the distinct invasive cell population or not. And then after that, we can use the 6-well invasion assay, transfer invasion assay, and then collect the invasive cells. And then for the second question, I forgot what was the second question, sorry.
So Ahmed was wondering if you tried to grow these, did you try to grow the invasive cells and characterize them after a few passages?
Okay. I actually grow the invasive cells and then proceed the passages, and then I found that their morphology or the doubling time, everything was similar to the whole cultured PANC-1 cells.
Excellent. Thanks, Mayumi. Our next question, and this one asks, have you ever tried this spheroid live cell imaging with other cell lines other than PANC-1, for example, MIAP-ACA2 or SF126? And are there any differences in invading population between each of those cell lines?
Okay. Yes, we have used CASCI, which is the cervical carcinoma cell line, and then MCF7, which is the breast cancer cell line. And then also we used SW480 for the colon cancer cell line. And then we captured their images for 3 days with live cell imaging with IncuCyte. But unfortunately, they didn't show that the distinct invasive cells population in that experiment. So far we confirmed that MIA-PANC-2 and then PANC-1 have the distinct invasive population. And from that information that we got from the live cell imaging, like we got the information that MIA-PANC-2 are invaded with the elongated shape with higher speed than PANC-1, and then in contrast to the PANC-1, to the MIA-PANC-2, PANC-1 used a rounded morphology to invade with a slower speed than MIA-PANC-2. And then in the case of SF126, it did not have such distinct invaded population like MIA-PANC-2 or PANC-1 did. But at the same time, like we got interesting information that cells placed at the edge of the spheroid actually move actively, but not move far apart from the spheroid like MIA-PANC-2 or PANC-1. So they didn't have the a distinct population of invading cells, but still they were like the edge of the cells were moving, but not going far apart from the spheroid. So for the answer of are there any differences in invading population between each cell lines? I think yes, there are many differences in invading population between each cell lines, and then we can only figure out by using the live-cell imaging with IncuCyte, then we can get a lot of interesting information and confirm whether they have the distinct cell population for invasive, phenotype or not.
Excellent. Thanks, Mayumi. And just a quick reminder to our live audience, we still have some time to ask a question. To ask one, all you need to do is type it in where it says type your question here and then press submit. So our next question, Mayumi, is from Sharisha, and this one asks, was genotype or metabolomic, sorry. So let me just start again on that one. So was genotype or metabolomic profiling or even further invasion assays applied to the propagated cultured invasive cells?
So can you say it again?
Yeah, sure. So was either genotype or metabolomic profiling applied to the propagated or cultured invasive cells? Was anything like that done?
I haven't done the experiment for the genotyping, so yeah, I don't have any answer for that.
Sure. Absolutely. No worries, Mayumi. I'm sure perhaps we can sort of follow up with that maybe offline a bit later. But let's move on to our next question. And this one asks, have you ever tried any animal experiments to examine the metastatic ability with using these distinct PANC-1 invasive population compared to the whole cultured PANC-1s?
Okay. That's a good question too. Thank you very much. Actually, yes, we are really interested in bring the invasive cells to the animal experiment, and then we have done the animal experiment as well. But I didn't show the data in this webcast because it is just unpublished data. So, but anyway, from that experiment, we have clarified that PANC-1 invasive population do have the higher metastatic ability to, especially to the liver, comparing with the whole cultured PANC-1. So.
Excellent. Thanks, Mayumi. Our next question asks, do PANC-1 invaded cells relate to the cancer stem cell? And have you ever tried any experiments studying whether PANC-1 invaded cells have stem cell properties or not?
Oh, okay. That's a good question too. Thank you very much. Actually, I have interested in that part as well, and then I also hypothesized that maybe PANC-1 invaded cells have some relation to the cancer stem cells. So I actually have done some experiments to figure out whether like invasive cells have cancer stem cell collectors or not. And then it's all unpublished data, so I haven't shown any of the data on my webcast slide. But I can say that like PANC-1 embedded cells did have some of the stem cell properties, but not for everything. Many, for example, many papers have reported that like CD44 is one of the cancer stem cell marker for pancreatic cancer. But there were not significant differences in CD44 expression between invasive cells and whole cultured PANC-1. But on the other hand, the ability of spheroidogenesis were significantly high in invasive cells. So some of the data shows that invasive cells have cancer cell property, but some of the data shows it's negative. So it is still, for me, it is still unknown whether invasive cells are the cancer stem cells or not. So I need further experiments for that.
Excellent. Thanks, Mary. And our next question asks, you mentioned that PANC-1 invaded cells exhibit resistance to oxidative stress. Do those cells show resistance to anti-cancer drugs or to radiation as well?
Okay. I haven't tried the experiment to see the response to the anti-cancer drugs, but I have done to see the difference in radio sensitivity, and then actually PANC-1 invaded cells are more resistant to some of the radiation types than the whole cultured PANC-1. So at least I think to some types of radiation, invasive cells are more resistant, and so they might have risk to survive more after such kind of radiotherapy. So from this point of view, it is really, I think that it is important to study and understand the characteristics of such invasive cell population and figure out how to block those cells effectively. Thank you.